Hello and good afternoon, friends. Welcome to the CEC-UGC live lecture. Dear friends, with another lecture on the series which we have started is food microbiology. Today we would be talking on microbiological safety of foods, and for this discussion we have once again with us in our studios Dr. Shalini Sehgal. Dr. Shalini Sehgal is Associate Professor in Department of Food Technology, Bhaskaracharya College of Applied Sciences, University of Delhi. Dear friends, let's welcome our guest Dr. Shalini Sehgal, under whose guidance we would be discussing on the topic microbiological safety of foods. Hello ma'am, welcome to the EduSet lecture.
Thank you so much. A very good afternoon to all of you. Today we'll be talking about microbiological safety of food. The topics which will be covered in today's lecture will be food safety, the hazards, the microorganisms, the history, the epidemiology, transmission, foodborne illnesses, and in the last, prevention and control. The entire talk will be in a two-part series where we will be discussing the introductory part first, and then we will be highlighting on the microorganisms which contaminate food and are a threat to the consumers who are having this food. So, whenever we talk about microbiological safety of foods, first of all we should know what is food safety. The definition of food safety is the food should be safe as per its intended use, or it should be eaten according to the guidelines which are given. For example, even common salt like NaCl will be toxic if it is taken in higher amount. When we talk about food safety, we also talk about conditions which are there while processing of that food, packaging of that food, and what are the various factors which are entering into that food during that. So, basically the definition of safe food is a product which is free of microbiological, chemical or physical hazards. So, before we start talking about a safe food, we should know what is a hazard. Hazard is a biological, chemical or physical agent in a food with the potential to cause an adverse health effect. In today's series, we will be focusing more on the microbiological hazards. Now, when we talk about the common food hazards, we can have hazards which are coming through bacteria, microorganisms, and we term them as microbiological hazards. Then we have physical hazards which are coming through small pieces of glass, metal which is in the food and which can cause various types of injury. So, they are termed as physical hazards. This is one category which can be easily tackled. The third category is chemical hazards which usually result in poisoning. What are these chemicals? These are the chemicals which are being sprayed on the food, which are put in the fields. These are the chemicals which are also used for cleaning the food contact surfaces in the food plant. So, they lead to poisoning and these are easily controllable. And the third category of the hazards which is upcoming nowadays is the allergic or the allergens. These are the hazards which are causing various types of allergies and they are coming through food, and the common ones can be the soya, it can be the peanuts, it can be various types of nuts. When we talk about the hazards, since the focus of today's lecture is microbiological hazards, so we will be talking about various classes of microorganisms such as bacteria, yeast, protozoa, mold, viruses. These all microorganisms, they make the food unsafe. And why this is so very important? Because you cannot see these microorganisms, they are all invisible. Therefore, they are a bigger threat to human beings or to the consumers of foods. When we talk about these microorganisms, there are various types of microorganisms like we have seen in the last slides. A total number of microorganisms which are capable of causing the diseases are around 250 in number. These microorganisms, they are coming through various sources and they are capable of causing the foodborne diseases or illnesses. When we talk about these microorganisms, the most common microorganisms which can cause the illness are the bacteria, followed by viruses, then parasites. So, in today's lecture, we are going to focus on the most important part of these foodborne illnesses which are the bacteria. Now, when we look at these microorganisms and we look back at the history, we can see human beings, they came to know early 90s that the food can be contaminated from various sources, and we can have contaminated water, we can have contaminated milk, and they were implicated in very many types of foodborne illnesses. Then there was a revolution. People started talking about cleanliness, sanitization, and there were new agents which were estimated, which were found out and were being used in the food industry. There were many things like sewage and water treatment, there were hand washing, there was a concept of hand washing, and people gave a scientific reason that the hands, they also act as a reservoir of many types of microorganisms which enter into our food, and hand washing should be a critical step because it is directly linked to a foodborne illness. Then people talked about pasteurization. It was in 1898 that Louis Pasteur talked about the concept of pasteurization, and it was in 1908 pasteurization was used in case of milk. After pasteurization, people understood not only heat but removal of heat, bringing the temperature lower, also can extend the shelf life of food, and there was the genesis of the concept of refrigeration. Refrigeration means if we are trying to keep the temperature of the food product lower than the room temperature, we can control the microorganisms, not only control the microorganisms but also extend the shelf life of that particular food product. This led to the concept of chilling, refrigeration as early as 1913. Now, when we talk of various types of diseases, these diseases, they are caused by food, and whenever there is a disease, there will be an epidemiology of that particular disease. How this disease is being caused, what are the reservoirs, which are the hosts, and what are the symptoms of this particular disease. But when it comes to food, many diseases, they go unrecognized, undetected, because most of the time we assume that these foods are cases of gastroenteritis. We never link a particular gastroenteritis case with a particular microorganism, especially in our country like India, where gastroenteritis is not given that kind of importance, and there are no trace back mechanisms that this gastroenteritis was caused by which microorganisms. We do not have any kind of inventory or a data which tells you about the food pathogens which are there in our country. Especially in different parts of India, we are having such a diverse agroclimatic belt. We have different types of people who consume different types of cuisines, but we do not have any kind of inventory on foodborne pathogens. There are pathogens not in food, there are pathogens which are there in water. Whenever we talk about food and water, we cannot talk in isolation, because now with the latest definition, water is part of food. You will not be able to make any food without the inclusion of water in any step. Now, when we are talking about pathogens, pathogens means disease causing microorganisms. These microorganisms, they can come from various sources, and even if we tackle these microorganisms, we will always find the literature which will say that there are emerging pathogens which are yet to be tackled. Why? Because microorganisms are, especially bacteria and viruses, they keep on mutating, and once they mutate, the earlier mechanisms which were there in check, they become useless for that particular species of microorganisms. Therefore, we keep on having emerging pathogens which need to be identified and which need to be profiled. Now, coming to the next thing, if we are talking about pathogens, and yes, they are going to cause disease, what are the risks? What are the groups which are at risk? So, first of all, the elderly people, because their immune systems are not very strong. Second category is the children who are gradually building up their immunity. And finally, due to third group, which are immunocompromised due to certain types of diseases. This group is increasing rapidly because due to various types of lifestyle diseases which are common nowadays. Now, when we talk about the transmission modes, the pathogens, they will enter the food through various routes. These routes can be the oral route, when you're consuming a particular food which is full of microbes, those invisible microorganisms, they're gaining an entry inside you. Second, the level of contamination will be different because this will depend upon the organism, the reservoir, who is handling it, the situation or the status of the processing, and cross contamination. For example, if you are having a raw food, it will be having a higher microbial load. If the same raw food is being processed, the load decreases. So if you eat something raw, the contamination level entering into your body are higher, and the processed food, same processed food from the same raw material, will have lesser microbial load, and your contamination dose will be lesser. There are certain instances where there is cross contamination. For example, when you are taking a raw food and you are putting a processed food next to it, there are chances of cross contamination, and we usually see that we should give different sections of the refrigerator for the processed food and different shelves for the raw food, so that there is no cross contamination happening. Always in the food industry or a good food processing unit, you will find that the design, the food should enter from one side and the food should leave from the other side. It should be a direct line or it should be a U. There should be no crisscross happening between the raw and the processed food, because it will lead to cross contamination. Then we talk about the human reservoir. Each microorganism needs a reservoir to multiply and grow, especially when we talk about viruses such as Norwalk viruses. These are very, very common. They are related to food in a great extent. Then we have Campylobacter, then we have Shegella. All these three microorganisms, these viruses, both bacteria, they are present in the human body. We act as a reservoir for them. If the human reservoir, why we are using the word human reservoir? Because we ourselves are going to contaminate the food if we do not follow personal hygiene. Now on the other hand side, there is a group of food which we say meat or related foods, animal foods. In that case also there are many types of pathogens. Why those pathogens? Because they are a reservoir in the animal. For example, Salmonella, E. coli, Listeria, Toxoplasma, they all are present in meats. So people who eat meat or related animal products, they are more prone to the outbreak, or they are more prone to diseases caused by these particular microorganisms. Now when we talk about sources of food contamination, there can be multiple sources of food, because food is grown out in the environment. It comes in contact with water, soil and air. There is nowhere on the globe where people are growing food apart from the environment for public use. Yes, for experimental purposes you can in-house grow certain types of food plants for some research purpose, but you see whether it is US, whether it is India, the food is grown out in the fields. Now when we talk about the food contamination, contaminants will enter from various sources, and if you see the slide, you can see they come from the raw materials, the ingredients which are there. They come from the water which is being used, they are coming from the air. Air is a major source, because in air we have dust and dirt particles on which these particular microorganisms, they ride on these dust and dirt particles and they settle down on the food. Soil, most of the plant foods are grown in soil, most of the plants are there which are, especially some vegetables like potatoes, which are the tubers or the root crops, they grow within the soil. Even the animals, they graze on the grass which grows on the soil. So soil is a very, very potent source of contamination, and many microorganisms, they come through the soil into the food. Then hygiene plays a very critical role. If you are not maintaining sanitation, and there are various types of sanitary measures, if they are not taken into check, they are not put into check, there are chances that the food gets contaminated. We have rodents, pests, this is again linked to your level of sanitation. If they are unclean surfaces, there are chances that they are going to breed the pest and the rodents. Insects are a very common source of contamination, especially the flies, and a single fly will have multiple or different types of microorganisms on its surface and is going to contaminate the food. Then we have animals and birds. They themselves act as a reservoir. We have already discussed in the last slide, but I would definitely like to focus on birds. For example, a bacterium like Campylobacter, it is very commonly associated with chicken, geese, poultry, because Campylobacter jejuni or pylori, it is found on the birds which are being consumed. Ducks, geese, chickens. So, this Campylobacter will enter into the food chain through these food sources. Then, now we are talking about processed food. If the packaging material is contaminated, then there are chances that your food product will also get contaminated, because as long as it is a manual packaging, then there are chances of contamination which are much more higher, which are much more significant in comparison to the packaging which is done by the machines. When we talk about the food and it is being packaged, there is a role of food handlers or the workers, because not all the food industries are fully automated. We have workers who are working there, and these are the people who are handling food at various stages of production and processing. When this is happening, there are chances that they themselves are contaminating the food which is being processed in that food plant. Whenever we talk about the processing of the produce means anything which is growing. It can usually is used for fruits and vegetables, cereals. This is the category which we put under the heading produce. There are various events which are happening during the processing, and they act as sources of contamination. So, I will be just taking few examples to explain you how they are contaminating the food. The very important event is the production and the harvest. When I use the word harvest, it can be picking it up, bundling it, putting into bunches, but this very event, which is very critical for the processing, will act as a source of contamination, because if we are using low quality irrigation water, if that groundwater is contaminated, automatically your fruits and vegetables which are grown in the field, especially vegetables, they will be affected. Rice is another example, because the paddy is grown in the fields which are full of water. So, if the quality of water is not good, that water itself, whether the metals are there, whether the microbes are there, they are going to leach into the rice produce. Then we have the kind of manure which is being added. We do not have very clear-cut instruction about the use of manure in our country. So, when we are using untreated manure in our fields, it leads to contamination, and usually the coliforms, the fecal coliforms, they enter into those food products. Because there is lack of field sanitation. We have no concept of field sanitation, the types of irrigation waters to be used, the kind of manures to be used. We have no clear-cut guidelines. Therefore, most of the time the food which is coming to us, for example, if I take Yamuna, any food which is grown closer to the Yamuna is heavily contaminated, because the water of Yamuna itself is contaminated. The next event which is very important once the food reaches to the plant is the initial processing. Whenever we talk about the word initial processing, means we are going to wash, we are going to dice, we are going to sort. In certain cases, if it is fruit, we are going to box them, we are going to put the whole food into the fruit into the box. So, there are chances that it will get contamination. Now, again the source will be the wash water, because again we are not focusing on the quality of wash water. Most of the industries, they are using groundwater for washing the fruits, then they are being packed and they are shipped to various places. The next thing is the distribution. We use the word trucking. We are loading our boxes into the trucks and we are sending it out to various places. Now, what happens in these trucks? There is no concept of supply chain or cold chain management. What temperature the produce is undertaking during the journey is not clear. There are lots of fluctuations, and by the time it reaches to the retailer or to the main hub, it gets contaminated. Then the final processing. Once that food reaches the home, people will be doing certain treatments like, especially in case of raw products like dicing, slicing, shredding, peeling. There are chances that it becomes contaminated, because the quality of water will always affect the quality of food, and water is a very, very important source through which the pathogens will creep into your food. So when we talk about all these exercises which are being done in home or outside, let's say eateries, canteens, cafeterias, there are chances that the contamination of the food will take place, even cross contamination, and the pathogens which were there already will start breeding or increasing in numbers. Now, since we were talking about the pathogens in food, and the focus usually is on the bacteria or it is on the viruses, and the difference between bacterial growth and the viral growth is one and the most significant. Viruses, they do not need a host to multiply. The viruses, they need a host to multiply, but the bacteria, they grow as such on the food. The growth mechanism of the bacteria is very, very fast. They just divide into two. The growth pattern is known as the binary fission, means from one we get two bacteria and so on. So within few minutes, you will have millions and trillions of bacteria which were earlier not there. So if you see, if we just take a common slide, which is this is a theoretical representation, from one bacteria within a span of 10 to 12 hours, you can have a billion bacteria. So this slide tells you how grave is the situation if it is contaminated, if we do not put checks into place, we do not take care of the food hygiene part, we don't talk about sanitation. Now, when we talk about the microorganisms, a very key factor is the temperature. The temperature is very critical because these bacteria, these microorganisms, they grow at their favorite temperature. This favorite temperature is known as the optimum growth temperature. Each microorganism will have a minimum, optimum and a maximum growth temperature. And to control these microorganisms, we have to use this very temperature as a tool. Whenever we talk about food safety or whenever we talk about foodborne pathogens, we talk about a concept that is a temperature danger zone. We divide this zone into three parts. The danger zone is usually from 4 to 70 degrees C, because this is one area this is one zone where most of the microorganisms will proliferate. If you want to keep a check on these foodborne pathogens, it's better that we keep the temperature less than 4 or we keep the temperature more than 70. Therefore, usually you will find a concept, keep cold foods cold, hot foods hot, because this will not permit the pathogens to proliferate and multiply. Now, when we talk about certain microorganisms in this particular lecture, I will be focusing on two common microorganisms. The first one is Staphylococcus aureus. As the name of this bacteria, Staphylococcus, Staphylo means in bunches, cocci means spherical. So anything which is spherical and in bunches is known as bunches which look like grapes, we call it as Staphylococcus aureus. Now, why I should be talking about this particular bacterium is that this is one particular bacterium which comes through the hands. The reservoir is human being. So, when we talk about S. aureus, food poisoning, these are the ones which are happening when the temperature is being abused, means we are putting the foodstuff outside for a higher temperature where this particular bacteria can proliferate. And why this bacteria is so significant? Because it produces heat stable toxins. Toxins which target the entron, and hence they are known as enterotoxins. And like I said in the beginning, why it is so very important? Because this is right here. This is on your head, on your scalp, it's there on your hands, and this is how it gets into the food and leads to acute cases of food poisoning. The other bacteria which I would like to mention is Bacillus cereus. This is ubiquitous in nature. The minute I use the word ubiquitous means present all over. This means if this bacterium is there everywhere, the chances of it entering into the food are much more higher. The percentage probability increases, and it leads to two types of poisonings. One is the emetic food poisoning and the other is diarrheal food poisoning. The name itself suggests emetic means lots of vomitings are going to happen, and in case of diarrheal there are chances that you are having loose stools or you are having diarrhea. Why? Because the toxin produced by this particular Bacillus cereus species targets your small intestine, and it produces a toxin which leads to the movement of water molecule, or it leads to the movement of salt particles out from your food and leading to various types of vomitings and diarrhea like situation. Then we have another member of this foodborne pathogen, Salmonella, which causes a disease which is usually known as gastroenteritis. But for most of the layman, people relate Salmonella with typhoid fever. Yes, Salmonella and typhoid are related. Typhoid is caused by Salmonella typhi, but when we talk about food and foodborne pathogens, we are talking about two other species. One is Salmonella enteritis and the other one is Salmonella typhi murium, and they cause various types of gastroenteritis. Now again the question, from where this microorganisms comes into the food? It comes from infected food of animal origin. For example, it can come through meat, it can come from eggs, it's also found sometimes in various types of fishes, but animal food is a source of this particular bacteria. So at this end, I would like to tell you, at this stage I would like to share with you that the foodborne pathogens, they will enter and they will cause various types of diseases. The routes can be many, like we have told you, but these routes, they are very, very critical if it is a human reservoir, if it is an animal reservoir, if it's a bird, then the chances of falling sick are much more higher, and we have to be very particular, taking care and controlling and minimizing their entry into this particular food environment so that the diseases are not caused. So thank you so much for listening patiently for this lecture. Good afternoon. Now I'll take you to the second part of the lecture, and we are dealing with concept of microbiological safety of food. Like in the earlier lecture, we understood about how microorganisms, they are gaining entry into the food and causing diseases. In this part of the series, I would like to share with you how they are attaching themselves into the intestine and causing various types of outcomes, symptoms, and usually we relate them with a disease or a disease state which is known as gastroenteritis. So for any microorganisms to act or to cause any harm, it needs to bind itself to the surface. So whenever we talk about microorganisms, I would like you to talk about a concept that in the intestine, most of the time we have various types of villi, villus. These are there to increase the surface area of the intestine, because main function of the intestine, especially the small intestine, is to absorb the nutrients. These villus not only increase the surface area, but they also provide the ground for the microorganisms to attach themselves to attach to these hair-like fibers, and they safeguard themselves from the various types of movements which are happening and various types of harsh environments which are there in the intestine. For example, when we are talking about a common bacterium like Salmonella, and we see it on the surface of various types of meats, and when we see it on the surface of meats, we talk about a concept of biofilms, because these microorganisms, they are living and they attach themselves onto the surface of the meat and they lead to the genesis of biofilms. These biofilms are not only formed on the meat surfaces, the same very biofilms are caused inside the equipments. If they are not cleaned properly, they are also formed inside the pipelines, and gradually whenever the food is being processed in these equipments or it is passing through these pipelines, it gets contaminated with these particular microorganisms. Now when we talk about Salmonella, and it is a microorganism which is commonly found in food, and it is implicated in most of the foodborne infections, and it leads to a disease which is known as salmonellosis. We talk about the sources. The sources of Salmonella are raw poultry and eggs. We can find Salmonella in raw milk, raw beer. It is also there on unwashed fruits and alfalfa sprouts. In our country, we are having various types of sprouts like moong dal sprouts, Bengal gram sprouts. There are chances that you can consume them and you can fall sick if Salmonella is present on those very sprouts. Now, what are the symptoms of this salmonellosis disease? It is going to take 12 to 72 hours of onset before you see the symptoms. So all of you must think, and you will realize that you have also fallen sick after a day or two after eating a food in some kind of a party or outside your home. The symptoms are not immediate. This makes food poisoning through chemicals and food poisoning through microorganisms different. Chemical poisonings, they show instant outcomes. People fall sick immediately, but when you fall sick by microorganisms, they will need an onset time, and this is especially in case of food infections where they will need 12 to 72 hours to grow and proliferate. Now this duration, why there is a lapse, why they take 12 to 24 hours or 72 hours to show the symptoms? Because symptoms are only possible when the bacteria reaches a particular level, number. So these 2-3 days are there because the bacteria is multiplying in a number. The minute it reaches its infective dose, you see the onset of these very symptoms. Coming to a very common microorganism, E. coli, Escherichia coli, every one of us is having E. coli in us because it is residing, it's a commensal in the colon of man as well as animals. Whenever we talk about microbes on the body surface or inside the body, around 3 kg of the weight of your total body weight is due to microbes, and these are the microbes which are living inside the colon, or they are on your skin, or on your scalp. And one such microorganism is E. coli. Why E. coli is such a common microorganism or a favorite microorganism of study? Because this is found invariably on all human species. This has been completely studied. Each gene has been decoded, and only few strains, some strains are pathogenic in nature and they are capable to cause disease. The diseases which are caused by E. coli are known as traveler's diarrhea or neonatal diarrhea. And why it happens? E. coli comes as a fecal contamination. Most of the time the water supply and the sewage lines are moving together in most of the cities. There are chances there is a cross contamination of this E. coli from the sewage lines, because after years there are chances that there are holes, pinpoint holes through which there is a cross contamination happening, and fecal contamination are also indicators of safety and quality, especially safety when we talk about food. When we talk about E. coli, there are various stages of pathogenesis of E. coli. How it attaches itself to the epithelial cell. We have fimbriae which are there. These are hair-like structures which help to the adhesion of the bacteria to the surface of the epithelial cell. The bacteria secretes a toxin, enterotoxin, and this toxin binds to the receptor on the cell surface and crosses the membrane. The minute it crosses the membrane, it modifies the regulatory protein in the cytoplasm, and if you see this diagram, you will find that the intestine has been shown and we have the microvilli. Intestine is lined by the epithelial cells, and you can see that this E. coli, which itself is having many hair-like structures which are various types of flagellae, and this is a peritrichous microorganism. It goes and lodges itself to the epithelial lining, binds to it, the toxin goes inside, and as a result, after a short time you will see that the water and the electrolytes, they start coming out. These water and the electrolytes, they come out from the human body in the form of vomits, or it comes out in the form of diarrhea, loose stools. This is one electron micrograph which again shows you hair-like fibers all over the surface of E. coli, and these E. coli cells are attaching to a villus or the fimbriae which are there. Now coming over to another microorganism of concern, this is known as Clostridium botulinum, one of very famous microorganism. Although it is found rarely in food, but whenever it is there in food, it produces a neurotoxin which can be very fatal and can lead to the death of the person. This particular microorganism, Clostridium botulinum, which is also known as Mr. Bot at times, produces, it is a neurotoxin producer. It is anaerobic in nature, so wherever people are having canned foods, there are chances that C. bot might be there, and it is a gram-positive spore forming rod. The minute you say spore forming, this means it is resistant to heat. So low temperatures, or you can say medium temperatures of heat processing, are not able to destroy this particular microorganism, and this particular microorganism also produces a toxin which is heat labile and is destroyed if heated at 80 degrees C for 10 minutes or longer. Now why this microorganism is so very fatal? Because it is not the vegetative cells which survive, but the spores which survive, and there are around 7 to 8 species of botulinism which have been identified. They are known as type A, B, C, D, E and F types, and out of which A, B, E and F are able to cause human botulism, means you can fall sick, human beings can fall sick after consuming C. bot, and out of these 4, B is the most commonest type to infect human beings. Now when we talk about botulinum, it leads to an attack, and it leads to a flaccid paralysis of the infants, because children, especially the newborn children, are always at a greater risk. And why? Because most of the households, even in India, as soon as a newborn baby is there, there is a practice of giving honey to those kids, and as a result it leads to various types of you can say poisonings, gastroenteritis, due to this toxin which is present in the honey which has been fed to the newborn child. Then there are chances that a person has various types of symptoms like drooping eyelids, difficulty in speaking, swallowing, and the outcome comes after 18 to 36 hours. Another microorganism is Shegella, and the disease is Shigellosis, and since Shegella is a rod, the disease is also known as bacillary dysentery. And the most common species of Shigella is Shigella sonnei, and it comes through human fecal contamination of food, beverages, vegetables and water. Whenever there is a disease, there is a foodborne disease, there are certain symptoms and signs. In this case, again we are having diarrhea, nausea, vomiting, cramps, fever. And how long you are sick due to this particular disease is 5 to 7 days. So now when we are talking about certain pathogens, I have shared with you some common pathogens, but the list is endless because daily we are adding new pathogens into this list. Now the question which should come to your mind is, why are there new pathogens? Why are we having emerging pathogens? There are many reasons behind the emergence of these new pathogens. Some of them are globalization of food supply. Now you can find food from different countries being sold in India, in Delhi, or various outlets across India. So when this supply is coming from various parts of the globe, those microorganisms are also find route to enter into our country. New processing and handling methods of food are fast emerging. For example, now if you see, microwave is a very common concept which was earlier not there. So what is the effect of microwaves on microorganism? That needs to be studied in detail. What species survive, what species mutate, and which still reside in the food needs to be taken into account. So with every new technique, we need to find out what is the impact of that new technology on microorganisms. Then when we are talking about adaptation, microbes are old, but they have adapted. They are resistant to the antibiotics which we take. Especially in our country where we have a rampant use of antibiotic, we are creating an army of antibiotic resistant microorganisms which are causing a threat to us. Then demographic shift. This is a very common thing that people from villages, they shift to cities for search of work. So the microorganisms which are common in one zone, one area, they gradually get diffused across areas in a particular country or from one part of India to another part of India. Then the other thing is changes in population susceptibility. As we read literature, we always see that we are having a longer lifespan. The age is increasing, the average age of human being is increasing, and there are new things which are coming up because their immunity goes lower and they become prone to new microorganisms, and there are new diseases which set in. Lastly but not the least is the changes in lifestyle. When we talk about the lifestyle, we are gradually a country where we used to cook food at home. Now gradually we are becoming a country which is eating food out. So this major shift has although added much more variety to the food, but it has also made us prone to various types of foodborne illnesses. Why? Because when you are cooking food within a confined kitchen, you can control many parameters. You can take care of hygiene, the sanitation level, but when it is cooked in a kitchen where you have no control, there are chances the standards are not met and you end up falling sick after consuming that particular food. Now, when we talk about emerging pathogens, I would like to share some pathogens which are fast emerging as Cyclospora. It is a protozoan which is there and which is commonly seen in imported raspberries. Listeria monocytogenes, it is a microorganism which has fast emerged as a foodborne pathogens. It comes through food, it can be seen in meat, cheeses, and it leads to human abortions and stillbirths. So, you can very well see it's not only gastroenteritis but also death, sometimes abortions, miscarriages happening due to microorganisms. It also leads to septicemia in case of young or low immune people. The another bacteria which I would like to mention is Yersinia enterocolitica. These bacteria, they are found in various types of food like raw milk, cream, oysters, vegetables, fish, poultry. Why they are such a big concern to us? Because they can grow at refrigerated temperatures. Because we all feel if we have kept something inside the refrigerator, it is safe. But yes, it can be safe, but it is not microbiological safe because there is a group of microorganisms which are proliferating at such low temperatures and they can cause a foodborne disease later on. Campylobacter jejuni, these are few names which are worth mentioning because these are fast emerging pathogens, and in our country we should be aware that from where these are coming and what kind of diseases they are causing. For example, again this is one bacteria which causes bacterial diarrhea, but on the other hand it can lead to a syndrome and even to acute paralysis. You can have paralysis and it can be due to this bacteria Campylobacter jejuni, which is entering your food through poultry, especially chicken. Now, when you talk about this campylobacteriosis, it is very important because it is usually causing various types of diseases, not only in human beings, but it is also infecting the human reservoirs are there, but it is also a reservoir, finds the reservoir in poultry, cattle, puppies, kitten, pet birds. Why I am talking about it? Some people they keep pets at home. So, this is one bacteria which comes through the pets and leads to various types of diseases like diarrhea, abdominal cramps, and people might not associate that their kitten or their pup is causing the disease, but yes, they are acting as a reservoir and Campylobacter is getting into your food gradually. Then talking about E. coli, we had studied in the first lecture that E. coli is a common commensal and not all the E. coli are pathogenic, but yes, I would like to mention one strain which is 0157H7. This is the most deadliest of E. coli and it is going to cause enterohemorrhagic strain. Why we are saying it is enterohemorrhagic? Because it will lead to bursting of the RBCs, hemorrhage in the entron, and it will come from various sources such as hamburgers, salami, lettuce, even juices like apple juice and unpasteurized milk, well water, and there are many sources like animals, cattle, mammals which carry this particular microorganism inside their body and it comes out in the fecal material. Now, whenever we are talking about E. coli 0157H7, it leads to a syndrome which is known as HUS, hemolytic uremic syndrome, and you can see it leads to acute kidney failure and it can be very life threatening. So, you will never imagine that I took water which was not good, which was not pure, or it was contaminated, and you can end up having a kidney failure. There is one more disease which is known as the mad cow disease, or it is in other terms known as bovine spongiform encephalopathy. The first case was in 1985 in UK, and we came to know about a new agent of disease which are known as prions. Prions are nothing but these are proteinaceous particles which are capable of causing the disease, and they cause lesions in the brain, and hence the name is spongiform encephalopathy. We are having viruses which are very, very important in foodborne pathogen, because these viruses, they keep mutating. And you will yourself see there are no cures for viruses, and up till now we haven't been able to have diagnostic tools to control these viruses, and these are spread from person to person contact, they are shed in human feces, they are also shed in the vomits, and they cause various types of diseases. The seafood, especially the shellfish, is a very common source of these Norwalk-like viruses and leads to foodborne diseases. Now, when we have talked about so many diseases, we should also know about the prevention and the control measures. So, when we talk about the preventions and the control measures, this is a very common concept in the food industry and very, very effective also, which is known as HACCP, Hazard Analysis Critical Control Point System. Now, why this system is there? This is there to monitor and put a check on various types of production processes. It also helps us to identify these food safety hazards and CCPs, critical control points. Because for any food which is being processed, not all the points will allow the entry of pathogens. There will be certain steps in the process which are going to take care of those foodborne pathogens. So, it is very, very critical for us to identify the critical control points through which the microorganisms will gain entry, because if we should put a check on these CCPs, and we can lower down the contamination or the risk associated with that particular food product. HACCP is usually used for meat, poultry, egg and milk industry. It is one system which should be applicable to all the food manufacturers or the food production processes, because this helps us in ensuring safety of food or safe food. When we talk about HACCP, we also talk about a concept which is fast emerging, although this is very well established all across the world, but in India now we have started talking about a concept, that is risk analysis. When we talk about risk analysis, it takes care of three types of it comprises of three steps. First is risk assessment, risk management, risk communication. Because in kind of food, which is a very, very dynamic system, you cannot only keep on doing risk assessment. You should find out how much is the risk associated with it, and you should have very logical scientific explanations. After that, we can talk about management. Once you know what is the risk, the step 2 is how to manage that risk, and rather than manage, I should use the word how to cut down that particular risk or how to curtail that risk. Then you will find a strategy to how to manage or to lower down the risk. Now, this strategy is useless if you do not communicate. So, the third pillar of risk analysis is risk communication. You have to tell the people, the stakeholders, the producers, processors, how they can manage that particular risk. So, you will see risk analysis as a very, very complex stage or a process which is incomplete if you do not do assessment, you do not do the communication, and then only we can say we can talk about a very effective risk analysis. In a country like India, we do not have risk assessment, we do not have the inventories to tell you what are the risks associated. Once an outbreak is happening, then only we start talking about the risk, the threat, and how people are affected and what are the control measures for it. So, coming to the end of the lecture, we should have an integrated food safety approach. Why integrated? Because when you talk about food, it's not only the consumer, it is the farmer, the farm level worker, the processor who is making that food product, the workers who are working, and even the consumer who is going to consume that food. They should know what is food safety, what guidelines, what precautions they need to take. Otherwise, that very safe food will become unsafe. So, we have to have integrated approach towards food safety, and it should have GMP, that is good manufacturing processes, good hygiene practices, GHP, HACCP, we have already discussed. Then we should have microbiological risk assessment. Like I told you, microorganisms are very, very critical to the safety of food. So we need to talk about the microorganisms. And finally, TQM. We have to talk about total quality management. It is an entire capsule, so that we are not dealing with separate entities in isolations, but we are integrating together, because that approach will only take us to food safety. Now, when we talk about various roles, consumer has a role to play. we have certain guidelines which we have already discussed, like prevent cross contamination, personal hygiene. We have to take care of the temperature control, because like I already said, there is a temperature danger zone. So when you talk about the temperature danger zone, you need to keep the microorganisms apart from that, means below 4 and below 7. So like this, we can proceed towards safe food, we can fight the bacteria, and we can keep the safety of the consumers and the foods in hand. Thank you so much.
With this note, thank you, ma'am. Thank you so very much for giving us a very, very productive session on the microbiological safety of food. And dear friends, if you have any queries, then you can mail us at info.cc@nic.in. We would love to answer your queries the next time when Dr. Shalini Sehgal visits our studio. Till then, take care. Goodbye. Thank you, ma'am. Thank you so very much.
Thank you so much.